Good morning, ladies and gentlemen. I'm very happy to present our work about the single-cell imaging, and it is also a very important platform we have developed in our university for investigating the gene expression and also the metabolic process in the single-cell levels. So this is the team member. Professor Young is also one of the key members of the team, and today I'm responsible for presenting this work. So this kind of platform is actually initiated when we are visiting Stanford University 2 years ago, and this is a team led by Professor Lei Singh, which is the department head of the diagnostic the radiation oncology department in Stanford University, and developed a platform which is called the radioluminescent microscopy, so which will be able to measure the heterogeneity of the glucose uptake in the living cells. So they make use of the radio tracers, and this is the design of the system in Stanford University. They using the scintillator plane, which is pasted under the culture medium, and then these are the cells, and they inject the radio tracer into the medium, and then the cell will taking up the radio tracer. For the FDG, they use the fluorodeoxyglucose as the radio tracer. So when the cell taking into the when the tracer taking into the cell, and then after the beta decay, then this it will the beta particle will be interact with the scintillator plane, and then the visible light will be emitted. So this kind of visible light is very, very weak in the intensity. So that's why they need to use the military use CCD camera, which are very sensitive to the low intensity light to capture the image. So they verify the signal. This come from the FDG. This is the standard clinical radio tracer for the PET imaging, and is correspond to the GFP signal. And this is from the ovarian cancer cell. So they also trace the time intensity profile of different cells, and you can see in this diagram, the yellow line is actually the signal from the FDG for every single individual cell, and then the red line is the average across all these cells, and the blue is the control, which is the signal picking up from the background. Okay, so they find that this kind of technique can visualize the tracer uptake at single cell resolution. So you can see different cells will have a different intensity profile, the time profile, and also the heterogeneity of this radio tracer, the 18F FDG uptake in different breast cancer cells. So this is the first finding. They identify the heterogeneity of the metabolic process, and also they verify with another fluorescent glucose analog, so which is called the 2-NBDG, is another fluorescent analog. When comparing with this radio tracer, when they overlay with each other, then they can see the correlated signal. So which and also we plot against the along this red line on the bright field. Then those signal FDG and the 2-NBDG is they correlate in a very large extent. So they prove that this is a very prominent approach to investigating the glucose uptake. So in our study, we established the same platform, but at this moment we using the fluorescent analog, which is 2-NBDG, the chemical structure, and this is the chemical formula and all for this analog. And this analog has been used to monitor the glucose uptake in the leaf cells and as an indicator of cell viability, and it can display the based on the excitation of this frequency of light, then they will emit the visible light, can be picked up by the camera. So, and also this is analog which is rapidly accumulating in the tumor cell when comparing with the non-malignant cells. So already proved in some article. And this can be used as optical markers for detecting the malignant cells. So this is a microscope we set up in our laboratory. So this is the microscope, okay, including the camera, which is a high resolution, high sensitivity CCD camera, military use, and also the laser exciter. So it's used to excite the fluorescent label, and also a system, okay, when we capture the data in fluorescent, in bright field, they can integrate together in this computer to a single video or multi-frame pictures. Okay, this is the protocol. We perform our experiment, the cell culture in this L15 medium, and the temperature, and that's also the other steps and incubation in the 24 hours, and then we align the cell cycle in the same starting point so that they can compare with each other consistently. And 1 hour glucose fasting for this kind of alignment. And also now after this fasting, we add the analog and incubator for one hour, and then we start taking the photos. And in total for 12 hours, because we know that the cell division will happen around 6 hours for the breast cancer cell. So we take every 5 minutes one picture for 12 hours and then group them together as a video. Okay, so this is the one of the picture in the video. So we find that when we start taking the video, we find that some of cells are round the cells and one is a spindle-shaped cells, but they are the same. They are the breast cancer cells. Okay, from this cell line. Okay, so we start taking the video when we find that the cell is in this according to the cell growth curve is in the log phase. Then we use it for the cell imaging. Okay. So we monitor in one single picture totally fertilized cells. Okay, each line here is a cell. At the very beginning, okay, the uptake of these 2 MBDG is quite low, and then they develop the profile, okay, time profile heterogeneously, and then eventually you see a large variation. And some of them are staying at a very low uptake stage, and some is to be taking more and then decrease, and then so we investigate them one by one, and then we find some interesting finding about the cell division. We find the cell 3, 8, and 17 they divide. For cell, this is should be 17 is a red line. It divide into 2 cells at 3 hours, and other in the green is cell 3 divide into 2 at 4 hours, and the third it divide at the about 6.5 hours. So no matter they divide at different time, but one of the characteristic is they have a substantial glucose uptake. It increase substantially and then it divide. And also the green line, you can see also the increase of the glucose uptake. So this we hypothesize that is a glucose uptake, substantial glucose uptake is necessary for the cell division. And this is one of the example for the cell division. So the cell become rounded, okay, and then take out a lot of glucose, and then divides into 2, and become spindle shape after the cell division. And also you see the light become darker. The glucose, they use up a lot of glucose during the cell division. And comparing with the before the cell division is a very bright color, and become lighter. So this is a video I prepared for showing this cell division. So it should be, I can remember, it should be this cell. So you see it becomes a round shape, very bright. A lot of glucose have been taken up and divide into two. Okay, and become darker and spindle shape. So you see the morphological change and also the change in the glucose metabolism. So, and also another finding, we trace the movement of every single cell. So from the first cell to the fertilized cells, and we trace the path and the total distance it takes. And we find that, so this is showing the path of how the cell moves for every single cell. And the total distance travel is the x-axis, and this is the uptake difference. So at the very beginning, the glucose uptake, and at the end, the glucose uptake, and we take the difference for each cell, and we plot every single point representing the data of one cell, and we find that the distance correlate with the uptake difference. Okay, so there is a significant correlation between these two. Okay, the conclusion. So in this study, we also observe the heterogeneous uptake in single cells, every individual cells, and also we find high glucose uptake before the cell division, and become lower uptake after the cell division, and the cell shape become wider compared with the spindle shape after the division, and the cell will also longer the travel distance. Okay, the migration distance, and it will uptake more glucose. Is also one of the findings. It will be related to the metastasis of the cancer cell. So this is just our preliminary finding, and what's next, we will using this live single-cell imaging platform to further classify the cell according to the metabolic feature. For example, the time profile of this glucose uptake, to classify the cells into groups. And then based on those groups, we can also compare the rate of the cell division. So would it be the one profile is the glucose uptake increase at the very beginning and then plateau will have more rapid cell division, or there's an increase at the very beginning and then drop will have a higher rate of division. We want to compare them according to this grouping, and also according to the grouping, we identify those candidate genes which related to the glucose uptake, and also we'll identify potential drug. And this could be not only personalized, but also based on the cell base. So we identify every single cell, we will use a different drug target on those cells, and identify genes for metastasis, because some of the cells travel longer, they have a higher uptake. Then we can also identify those genes related to those metabolic pathway to target those genes to against the metastasis. Okay, so that's all. Okay, thank you.
So the floor is open for questions. At the back.
It's impressive work. I just want to ask, haven't you thought about resetting all your cells or aligning all your cells in the same stage of cell cycle before going through these classifications?
Yeah, in the protocol, we have already aligned those also cells after the glucose fasting. Yeah, so I think I have mentioned here. So the cell cycle should be, yeah, fasting.
So just the glucose fasting, you
Align it to the same, yeah.
So why you use Plaza bottle conditions?
Sorry?
Is it recommended for this type of cell line, or it's related to uracils?
Oh,
Plaza, not polystyrene, ordinary polystyrene bottom conditions.
sorry,
Yes, that's B. You use glass tissue culture dishes rather than plastic tissue culture dishes. So why would you use glass and not the plastic one? Are the cells adherent to glass but not?
I think that will be one of the, because
it specific to this cell line?
yes, if you use the plastic, then I think the adhesion will be changed, the adhesion property. So we want to demonstrate.
For migration purposes.
Yeah, for migration. So at the very beginning of the migration, they become the round shape, and then they detach from the bottom of the dish, and then it can migrate. So if you're using plastic, I don't know whether this kind of property will change.
Is it also that glass is better for imaging as well?
Yes, yeah, for fitting this kind of microscope, the thickness of the bottom will also.
I like your idea about using just smallest fluorescent dye, which is NVD's smallest fluorophore. You have quite high background of the fluorescent glucose when you have it emitted. But did you try to use caged fluorophores for your say?
Caged. I don't know what's cage. Yeah.
Caged fluorescent that basically a lipase activated. It's only activated inside the cell.
Not the that kind of laser excitation.
It's available.
Yeah, we can consider that. Yeah, but also the software allow us to remove the background. I think that would be.
This is very suitable for high throughput screening.
Yeah.
Very good to see.
Yeah, and also later on we have already purchasing the single molecule imaging system. Is based on the total internal reflection technology, and this kind of problem can be solved and more specific for using that kind of microscope.
I had one other question as well, and that had to do with a rather interesting graph that you showed, where you showed cells that went into division retained their glucose uptake characteristics after division as well. But it looked like those that had high glucose uptake and then divided, their daughter cells also had high glucose uptake.
Mm-hmm.
Those that had low glucose uptake and divided, they also retained their low glucose uptake. So, as you take a look at your cells, do they change their uptake characteristics over time, or do individual cells and their daughter cells and granddaughter cells, do they all retain the same characteristics? In other words, is there a stable difference between cells in terms of glucose uptake, or the dye uptake that you have, the glucose analog?
Yeah, I think this will be a very interesting question we need to answer in the future.
Because they change all the time. You may not be selecting individual subpopulations. And one very interesting question would be, do those cells that do not undergo division, which may be stem cell populations, do they have the highest glucose uptake? And the final question I would have, which I don't think you can yet, would be in vivo. What happens in vivo? Can you use this as an in vivo measure? Glucose analog?
No, but this is having a comparable property with the FDG. So FDG is a clinical. So that's why the next step we will also apply the FDG. Yeah. So that will be can be applied readily applied to the in vivo.
Thank you.
So what's the difference in the mechanism of the FDG versus the in vivo? Because the fluorescent one, is it only looking at the root receptors, or is it looking downstream for?
Yeah. So maybe I can show you this. Yeah. So the difference is the setting of the microscope. They need a scintillator plane to convert the signal for the FDG, convert the signal to the visible light. So 2 NdBg is fluorescent signal.
I'm asking biochemically.
Oh yeah.
So usually glucose comes into the root receptors, right?
Yeah.
And then for FDG, it gets phosphorylated.
FDG, yeah, but it will not generate ATP.
Yeah, but
after that.
Phosphorylated by hexokinase, so it doesn't go downstream.
Yeah, it's go downstream.
Does the fluorescent dye, is it looking at glut uptake, or is it looking at downstream of glut uptake?
As I can remember, it's similar to FDG. It's not no downstream. It's just trapped in the cell.
So it's getting phosphorylated.
Yeah.
By hexokinase?
I need to check whether it's a phosphorylate. Yeah.
Okay, because then you could be measuring different parts of the group of the glycolytic pathway by different
Yeah.
methodologies. And it would be telling you something different.
Yeah. Okay. Uh,
Because right now, the theory for FDG is that it looks like at the root receptors more than it looks at hexokinase.
mhm.
So it would be interesting to see what the correlation between your fluorescence and FDG.
Even a co-registration of the FDG uptake plus the dye uptake at the same cells.
Yeah.
Very interesting to see.
Yeah.
Thank you for your question.